2, 1.
Good morning, everyone. Welcome back to the second session of day 3 of our Hypoparathyroidism Association conference. I had a lot of questions regarding our Twitter and Instagram accounts. So if you could see on your screen, those are our Twitter and Instagram accounts. So please be sure to follow us. Also reminding you, your microphones will be muted, but you do have an opportunity to ask questions on the right-hand side of your screen. Now, I will present our next presenter. It is an honor to welcome Jamie Greene, who is co-founder of the Hypopara Research Foundation. In 2018, Jamie's ex-wife, Amanda, was diagnosed with hypoparathyroidism. Oh, I'm sorry. He founded the Research Foundation in 2018 after his ex-wife, Amanda, was diagnosed with hypoparathyroidism in 2016. Previously, Jamie spent more than 10 years in finance. Jamie graduated from the University of Pennsylvania with a degree in political science and has spent more than 10 years working in finance. He co-founded the Hypopara Research Foundation to bring global hypopara community together to fund research with the ultimate goal of curing hypopara. You can find out more on his website, which is hypoparatfoundation.com, and please be sure to visit him in the Expo Hall, which is on the left-hand side of your screen under the Teams tab. So without further ado, here is Jamie Greene. Thank you, Jamie, for joining us this morning.
Thank you very much, Barry, for the introduction. I apologize in advance as the new normal in Zoom calls if my children come barging in or my dogs start barking. First off, Barry, thanks. And then secondly, I want to thank Deb Murphy and the rest of the Hypoparathyroid Association board for inviting me to speak this year. The last time the Hypopara Research Foundation publicly spoke was when my ex-wife was the keynote speaker at the 2018 Hypoparathyroid Association conference dinner. It was the first time I had met anybody other than my ex-wife with hypopara, and it set the stage for the last 2 years and my driving motivation going forward. We certainly got some crazy looks when she announced at the dinner that we were going to raise $1 million in our first year and start marching towards a cure. We are happy to report that we raised the money and are marching towards a cure. And special thanks to the Hypopara Association, which recently gave us $30,000 out of their research fund to go towards research that we're sponsoring. So in this time we have together, I am going to give an overview of our foundation and review the 3 grants that we gave in 2020. Next slide, please. But first, I want to start with our mission statement. Our mission is to fund innovative, collaborative research with the narrow aim of improving the quality of life of those suffering with hypoparathyroidism and ultimately finding a cure for hypopara. Hopefully that clarifies some of the difference between the Hypopara Association and the Hypopara Research Foundation. The association is focused on patient advocacy and puts on amazing events like this, while we are focused on raising money and getting it into hypopara research. Next slide, please. I think it is important to give the background of my story and how I came into the hypopara world. My ex-wife was diagnosed with post-surgical hypoparathyroidism in 2016. The first hospitalization was an experience many of you have probably had, and it was the scariest day of my life. As I went from having a conversation with her to her being unable to talk or walk in about 5 minutes. That was the start of many ER visits for her. Unfortunately, it is a story that many of you can relate to. I soon learned of the plight of hypopara patients, the endless amount of pills, brain fog, extreme fatigue, tetany, inability to walk around the block, and countless other symptoms. Suddenly your life turns very, very limited. Family and friends become confused with you as you look fine, But sometimes you're really handicapped. You can't get out of bed. You can't, at a last minute, you have to cancel an engagement you had. The swings are so hard to predict, and a fear of any socialization takes over as you worry about the next crash. Lurking behind all of this is the debilitating trepidation in the back of your head that the next crash will take you to the ER. Whereas we've heard this weekend, the doctor might not even believe you as he slowly takes his time since he thinks you are having a panic attack or just seeking drugs. Ultimately, the Hypopara Research Foundation was started because we needed hope. My ex-wife was in her 30s when she got Hypopara, and we decided we had 50 plus years to cure the disease. So we started getting to work in late 2018. We had hope. Next page, please. Next slide, please. Thank you. The sole purpose of the Hypo Research Foundation is to raise money to make grants that will fund research to prevent, treat, and cure hypoparathyroidism. We think we are fairly unique in that 100% of donations go to research. All expenses that are incurred are covered by the board. Additionally, we only have volunteers and no salaried employees. The many people who have donated to the foundation know that every dollar they give goes to grants, and we are very proud that 14 months after forming, we were responsible for raising $1.2 million that went towards these 3 exciting grants that I'm going to talk about. Nobody in our immediate family has any relevant healthcare experience. I had been working in finance, as Barry mentioned, and I knew I wanted to start a foundation, but I didn't know where to begin. I didn't know how to get into research. I eventually got connected to the Orphan Disease Center at the University of Pennsylvania, and it quickly changed the trajectory of our foundation. Next slide, please. Full slide didn't populate, but that's okay. In the United States, a rare disease is characterized as affecting fewer than 200,000 individuals. There are over 7,000 identified rare diseases, with 1 in 10 Americans having one. Rare and orphan diseases are used interchangeably. With these kinds of statistics, it's clear why these diseases were called orphan. Orphan because the pharmaceutical industry abandoned them, because there are not enough patients in the disease to justify the cost of market development. So many patients were abandoned and excluded from the pharmaceutical industry and excluded from even the hope for desperately needed treatments. These are patients who need help the most, and oftentimes these would be the same populations who will create the lowest financial outcomes. Ironically, many of these diseases may be the easiest to treat with novel therapies, which we will get into shortly. Oh, there it goes. The rest of the slide popped up. The Orphan Disease Center facilitates and manages therapeutic development for rare diseases, with a primary focus on extremely rare and underserved patient populations. Given there are about 75,000 Hypopara patients in the US, the Orphan Disease Center was eager to work with us. We feel extremely lucky to be working with them. They are a very impressive organization. As you can see on the slide, they have funded $27.2 million in research since 2011. They award about 60 awards in 30 plus diseases annually. They are international in scope with multiple international RFAs and grants to over 100 institutions. They are providing so much hope to so many people like us in the Hypopara community who didn't have hope, who didn't have anybody in their corner pulling for them. Jim, who you can see on the left, is the director of the Orphan Disease Center. He is also a pioneer in the field of AAV gene therapy, a technology platform designed to provide a corrected version of a defective gene. And this has been very useful in some rare diseases. His fame gives a lot of credit to the Orphan Disease Center, and it allows them to collaborate with some amazing people. Monique on the right is the executive director of the ODC and works on patient outreach, research, and grant programs. They have both been intimately involved with us at the Hypopara Research Foundation and have gotten us to where we are today. Next slide, please. This is a quick slide which summarizes what the Orphan Disease Center, or ODC, does to help groups develop research for their rare disease. We are one of the first diseases to go through the Jumpstart program. Next slide, please. After initial outreach to the ODC from us, the Orphan Disease Center quickly put us into its Jumpstart program, which was then, we were one of the first groups to go through it. And it has been really instrumental to our success, and what we have done is being replicated in other rare disease groups. They helped us organize a key opinion leader think tank in January 2019, and shortly thereafter we joined their pilot grant program with 3 initial grants, 2 stem cell consortia and 1 device grant. Next page, please. So quick background on this key opinion leader symposium. In January 2019, our research foundation, in collaboration with the Penn Orphan Disease Center, sponsored a first of its kind event, a 2-day hypoparathyroidism symposium with experts from around the world. It was truly an international event. We had 30 participants, including doctors, researchers, investigators, pharma representatives from 4 different continents. This was truly our coming out party with the medical, pharma, and academia groups that were focused on hypoparathyroidism. Discussion ranged from drug companies sharing data that hadn't been publicly shared with each other, to discussions on novel therapeutics, to panels on stem cells. It was really an incredible event with researchers who had heard of other researchers for decades in their small field of hypopara, but had never met each other before this conference, which was amazing to me. It was an incredible 2 days where people could share ideas in a very closed environment. So ideas were very free flowing. On our website at hypoparatfoundation.com/happeningnow, there's a link. You can see all the participants, the full agenda, and summaries of each presentation. The one thing that I tell the Orphan Disease Center as a California kid, I definitely think the next symposium will not be held in Boston during the winter. But that's go on to the next slide, please. Thanks, Barry. So when the Hypopara Research Foundation was formed, we thought there were 3 places we could direct money towards research to help. 1 was to develop new drugs, 2 was in stem cell research, and 3 was in devices. We looked around and saw that there was healthy competition amongst the pharma company, which was great to hear from them yesterday, by the way. And we also realized it was very expensive and hard to develop drugs. With our limited money, we decided that initially the foundation would focus research on stem cells and devices. We partnered with the Penn Orphan Disease Center to fund 3 research grants in 2 critical areas: long-term disease treatment via stem cells and short-term quality of life improvements via a device. For a small foundation like ours, the ODC was invaluable. They instantly acted as our scientific review oversight board. We told them very high level, and might I add, not very scientifically and technically, that we wanted to fund a stem cell project and an in-home calcium monitoring device, and they took it from there. A program manager from the ODC managed the entire process, including writing the RFA, distributing it to the international research community, and reviewing each proposal through a blind peer review, and recommending to us which proposals to grant money to. I want to note the importance of this blind peer review, which I highlight in this selection process at the bottom of this page. We wanted to eliminate any bias that the Hypopara Research Foundation had. We had interacted with many of the doctors who applied for grants at the symposium, and Amanda had actually been a patient of several of the doctors who applied for money. So for the stem cell grant, Penn Orphan Disease Center brought 2 stem cell experts and 2 that's Hypopara experts to review each stem cell proposal we received. So they blindly graded each proposal and gave it a number score, and then based on that score recommended who we should invest our dollars into. Same thing for the in-home calcium device, 2 device experts for this, and then 2 disease Hypopara experts. So based on the recommendations, we were able to grant money in a non-biased way. Next slide, please. There are going to be a lot of big words in the next couple of slides describing the work done by each of the 3 groups. Again, I'm not a technical person, so I'm very glad that Dr. Krause went before me for some of the words in stem cells. So hopefully you got a better picture of some of the work we're trying to do in conjunction to Dr. Krause. But coming from a non-technical background myself, I will do my best to use words so we can all understand the research. If anybody has any questions, please let Barry know, and I will try my best to answer. So you can see here our three grants. The first was to Dr. Meyer's group. We granted them $500,000 in the first year. He is working on a stem cell project. The next was to Dr. Mannstadt's group. I hope everybody was able to hear him speak. We granted his group $500,000, and the next was to Dr. Wang, and that was a $200,000 grant to work on the at-home calcium monitoring device. We are really proud of these groups. If you look at their bios, if you look at the schools they've attended, it's an impressive group that's working for all of us. Next slide, please. First, before I get into this page, which has a lot of big words in it, I want to talk about stem cells at a very high level. The way my non-technical mind thinks about stem cells is it's a cell with the unique ability to develop into specialized cell types in the body. Think of a human. We all started as a single cell. That's right, one cell. That cell eventually multiplies and becomes specialized cells throughout your body. Stem cells are so important to research as scientists are learning how a cell can develop from a simple cell into a specialized cell with a specific job function. I thought Dr. Krause did a great job at describing that, and if people are looking at this after the conference, they should look around the Hypopara Association website to see her presentation on their amazing stem cell work. Recently, in the stem cell world, there's been the development of iPSCs, and these are induced pluripotent stem cells, which are man-made stem cells that are essentially formed from a piece of skin tissue of a human and deprogrammed back to a nothing cell. These man-made stem cells that can be programmed to potentially become any cell or tissue in the body. For us, the goal is for these nothing cells to become parathyroid cells. When we dive into Dr. Meyer's research, he first needs to fully understand how a normal parathyroid gland develops in humans to be able to transform stem cells into parathyroid cells in the lab. His ultimate goal is that these cells made from iPSCs can be transplanted into humans to replace missing or damaged parathyroid glands, and hence restoring production of parathyroid hormone. That's our ultimate goal at the Hypopara Research Foundation, is to cure this disease. And right now we think stem cells is our best chance, which is why we made 2 grants to it in our first year. Taking a step back, if you really think about it, it's remarkable that humans have created iPSCs. Dr. Krause described this earlier, but I'm trying to do this in the way that makes sense for me. And when I think about my body, from the stomach to the neck is an area called the definitive endoderm, and it is composed of major organs like the lung, stomach, and liver. As you work your way up to the neck, it is called the AFE, and this is where the parathyroid and thyroid develop in our bodies. Scientists have been able to recreate liver cells in the DE region, which leads scientists to believe that as their knowledge expands, humans should be able to recreate cells from the AFE region, similarly to how a baby develops cell, or how a human develops from a single cell and then expands up. They think this is very possible for parathyroids, but not enough money or time has gone into it yet. Next slide, please. So in Dr. Meyer's first 6 months, they've developed a computer software that will help them better understand how cells differentiate and become parathyroid cells and how genes are regulated in this process. The two mouse models they have developed have a parathyroid dysfunction similar to humans. And then once iPSCs are converted into parathyroid cells, they will be engrafted in these mice to test their functionality and unwanted side effects. The first paper by Dr. Meyer's team that cites the grant from the Hypopara Research Foundation can be found on our website. Again, you can go to our website, hypoparafoundation.com/2020 grant recipients to see that link and read that paper. Next slide, please. Next, we have Dr. Mannstadt's group, and he is also working on a stem cell project. And honestly, his objectives are very similar to the previous grants and very similar to Dr. Krause's work and her team's great work. First, all three groups are trying to understand how a normal parathyroid gland develops into humans. How do we have this thing called a parathyroid that not only produces the PTH hormone, but it also regulates us in it? Next, we need to be able to transfer stem cells into parathyroid cells in the lab. And third, we need to transplant these cells first into mice, eventually into people, to restore production of parathyroid hormone. The Mannstadt group will encapsulate the cells in tiny gel beads, inject them into the mice, and study the secretion of the hormone, while the Meyer group will graft and implant the cells in the mice. I don't want to speak out to turn, but so Dr. Mannstadt will have to confirm this. But in response to a previous question in Dr. Krause's presentation about the body attacking the new parathyroid, it's Dr. Mannstadt and his team believe that their encapsulation procedure would actually protect against any immune attacks, so people would not need to take rejection medication, which is really, really exciting. Okay, so why did we decide to fund two groups doing very similar work? Well, we wanted to improve the shots on goal here. We think the more people we have working on this, the more likely that one of them will succeed and cure hypopara patients. I can't say it enough. I was thrilled to listen to Diane's presentation on stem cells. It's more shots on goals for the community. It's more hope. We are really lucky that we have multiple groups working on this. There have now been several different groups who have written about their lab's ability to differentiate, make these parathyroid cells from iPSCs. I was so hopeful that that Diane's work was able to be reproduced by Dr. Levine's work. This replication of her finding in other groups' labs is very important. Stem cell research is still early. There are some clinical trials, not for parathyroid, but for across the spectrum in terms of stem cell use, and there have not been a lot of positive results yet. That being said, I really believe, and a lot in the Hypopara community believe, that Hypopara is the most likely application for success with iPSCs because you need so few cells to be transplanted in order to have a physiological effect. Think about it this way. The liver is 50,000 times larger than a parathyroid gland. So you would need so much more for a liver application to be successful. The small size of a parathyroid makes it a really good model. Next slide, please. So here we have our 6-month update for all our grants. We gave them at the beginning of 2020. Obviously, with COVID, there has been some delay, but all 3 of our groups are continuing to work throughout COVID. So Dr. Mannstadt's group have made progress in identifying important genes in the development of parathyroid cells using mouse embryos. They have also made some technical progress in the way they screen for cells and also the way they test their cells for markers, improving the overall protocol to make parathyroid cells from iPSCs. They've also developed a method to trap small fragments of parathyroid tissue into these microcapsules that I talked about before that are made of gel from seaweed, and they plan to transplant these beads into mice, which is very exciting technology. Go to the next slide, please. Here is a picture of the Mannstadt team. Thank you to them. Thank you to Dr. Meyer's team, to Dr. Krause's group for giving us all hope. These are the people who are working tirelessly behind the scenes, trying so desperately to help us. They realize what the Hypopara community is going through, what patients go through, and we are so lucky that they are trying to solve this for us. They are truly giving us hope. Next slide, please. So next we get to Dr. Wang at VCU, and they are developing a device similar to those used by people with diabetes to measure their blood glucose levels at home. The device should be easy to use, safe, low cost, and accurate. Hopefully everybody was able to listen to Liz from the from the Parathyroid UK speech on the first day of the conference, give her exciting update about Tom Kurzewski, I apologize if I pronounced that wrong, and his promising development of an at-home calcium monitor. More hope for the community that help is coming. At this stage, people with hypopara rely on self-regulated management. Imagine having diabetes in 2020 without the aid of a glucose monitor to guide treatment at home. That's what all hypopara patients deal with daily. And all of you on this call know the difficulties of going in and having to give labs when you're starting to feel terrible. And the ER doctor saying, hey, your levels look fine. And you say, yeah, that's because I just popped 6 calcium pills. I'm glad they're fine. You know, you're getting your results 3 hours after you were feeling bad. It really hampers management of your disease. Just for reference, 10% of the USA has diabetes, and at-home glucose monitors have been around for 40 years. 1980 was when the first at-home glucose monitors came into production. The Hypopara community should not be 40 years behind. The technology exists, just no medical device company has taken the time to put it into production. Not only does the technology already exist, but a machine already exists that could test calcium levels at home. It is called an iStat machine and costs over $10,000. Based on all the other tests it can do. It's very frustrating. It's a great device, but it's primarily used in emergency rooms for quick blood work by doctors. We are not trying to recreate the wheel here or what Liz's team and Tim is trying to do in the UK. We are trying to convert existing technology in a Ferrari, which I'll call the iStat a Ferrari, into what we need, a calcium monitor, which I will call the Chevy Spark, which I Googled last night, and it is the cheapest car right now in the USA. So we need to turn a Ferrari into a Chevy Spark, and that's what hopefully we can do soon. Can you flip to the next page? Thank you, Barry. The 6-month update is very promising for Dr. Wang's research. They have developed a prototype and have been performing initial blood tests on people. Excitingly, they only need 15 microliters of blood for their testing to work. By comparison, a drop of blood is 50 microliters. So they need 1.5 and a drop of blood is 5.0. Ultimately, here the goal is to have results hooked up to a smartphone so you can quickly share the data with your doctors. All of you listening, I hope, can imagine how life-changing that would be to get your calcium levels in real time and be able to share it with your doctors. The current focus for this group is to continue testing blood from patients to refine accuracy and sensitivity. Next slide, please. That's a quick recap of our first 3 grants so far. And I always go to what's next for the Hypopara Research Foundation. How do we want to spend the time or spend the money that we're raising on what sort of projects? Obviously, we'd love to continue the stem cell research. In year 1, as I mentioned before, we decided to fund 2 groups to do similar work to improve the shots on goal of success. There are multiple groups around the world working on stem cells for Hypopara. There's been stem cell reports written in Korea on Hypopara on parathyroid stem cells. You have Dr. Krause's paper. You have the couple guys presenting, or a couple guys we've backed. There's investigators in Europe working on it. It's really an exciting time in stem cell and the potential there. More shots on goal, better for us. More hope that something actually works. Next, we want to commercialize an in-home calcium monitoring device. We want to work with a group to get something to market. We are not doing research for research's sake. We want to create something that will actually help you immediately in this case of an at-home monitoring device. Next, we want to continue hypoparathyroid think tanks with experts from around the world. We would ideally love to hold those every couple of years, allow people to get in the same room, talk about what their research they're working on, talk about what others are working on, and really learn from each other. Next, we really want to study pump technology to make it the standard of care for hypopara patients. An NIH study from 2008 shows that pump therapy gave the best clinical results in children and adults, as well as the best kidney protection. Anybody who has been to a previous Hypopara Association conference has been intrigued by the handful of patients walking around with a diabetes pod that these patients have begun putting their PTH medicine into. Why is this not the standard of care? It is very frustrating to me. The constant small dosing of a pod, of a diabetes pod, closely mimics normal body function of what the body does with getting a PTH hormone. Getting a shot of PTH, boosting up your PTH hormone, and then slowly coming down throughout the day makes many people feel awful. They do avoid the drastic crashes that lead to hospitalizations, but it's not that calm, steady deliverance of PTH hormone that is best for the body, proven by an NIH study so far. We also, a couple people have mentioned over the course of the week, exploring parathyroid mapping projects. It's exciting new technology. Single cell sequencing combined with mapping the location of every individual cell type in the parathyroid gland will give a great base layer level for researchers going forward as they study the parathyroid and learn more about it. All right, we will flip to the final slide of my presentation. How can you help? Are you interested in volunteering, fundraising, social media coordination, website design? Please contact me, Jamie at hypoparafoundation.com. As I said, we are a small group of volunteers right now, and we could use anybody's help. So if you have any special skill that you think would be useful to us, please reach out. Even if it's just fundraising in your community. The Hypopara Association needs money so they can put on events like this. Please donate to them. Please donate to us. Get your family involved. Really, please consider donating so we can continue this vital work. We need to raise $1 million to continue our 2 stem cell projects in 2021. We are about 25% of the way there. We need even more money if we are going to do any of the other projects on our wish list. Please talk to your family and friends or companies who will do a corporate match. Thank you, everybody, for listening. Thank you to everybody who has already donated, which has given so many people hope. Barry, I'm now happy to answer any questions people might have in our last 7-8 minutes. Barry, you're on mute. Maybe it's we don't have any questions.
No, you have questions, Barry.
Okay.
You're just on mute. There you go.
I am muted. Here I go. Thank you, Jamie. The work you're doing is amazing, and we really appreciate it.
Thanks,
A couple of questions here. Someone wanted to know if the work that the UK is doing with the calcium monitor, is it all related to the calcium monitor that you are working on or hoping to work on?
It is Tim's work and our work are, and the work we funded are different projects. Same goal. So from our perspective, we are agnostic. Who gets the first calcium maker? I don't care. I just want it in the community as fast as possible. So we were thrilled by the update from Liz on Tim.
Now, is there a way that we can volunteer to have our blood tested?
Right now, it's just Dr. Wang is in VCU. So if you're in Virginia, that would be helpful. But we're not doing it, or his group is not doing it across the country at this point. As we go to commercialization, we'll need more people. That is why it is so important to sign up with the Hypopara Association so they can have a registry of patients, sign up on our website so we can have a registry of patients. So when we need people, when pharma companies need people, when device makers need people with our condition, we have people we can outreach to.
Now, will the device measure serum calcium or ionized calcium?
Serum calcium.
Serum calcium. And now, how much do these groups need to continue their research? Do they only get funding from you, or are there outside sources?
That's a good question. They try to get as much funding from as many different sources as possible. We've heard the news that the NIH has cut back a significant amount of funding, which is devastating to orphan diseases, which already don't get enough attention. So the government in the US has cut back on funding. Our hope with these first 2 stem cell projects was in year 1, they would give an update presentation to the Orphan Disease Center and to us at the Hypopara Research Foundation. If the Penn Orphan Disease Center thinks they've made enough progress, thinks they're moving in the right direction based on their initial proposal, we would like to fund each of those groups another $500,000 into 2021 to continue for another year on their work. Again, we've not guaranteed anybody that money. We want to see that their research is working. And if there's other groups out there that we think have jumped the line, we will go fund those groups. We are just trying to be at the forefront and getting money into groups that are moving things forward. Same thing for the at-home calcium maker.
And how far off do you think they are with that at-home calcium monitor?
Well, as I said before, the technology already exists. There are a few patients I've actually talked to in the Hypopara world who have iStats at home and can monitor their calcium levels in real time. This isn't new technology. It should have, this has been around for 40 years since they've been able to do it in glucose. And for diabetes, it's the exact same test that we need to test for calcium in our blood. So I don't think, I'm not a scientist, but we're not recreating the wheel here. So hopefully sooner rather than later. The time is not to create the technology to be able to do it. It's to be able to get the approval from the government, have somebody take on the product, a device maker, and bring it to the public.
Okay, now a question came in. Now, I don't know if this would be a question for you or maybe
if it's very technical.
It's not technical. It might be.
Email me if it's very technical, and I will reach out to the researchers to get you the full answer.
It might be a better question for Ascendis, but because it has to do with the TransCon PTH. But does TransCon PTH technology do what the pod would do, continuous delivery of PTH? I know that it's
I don't want to speak on what TransCon is going to or not going to do, but certainly excited by the initial findings that they've released to the public there.
Okay, now we have two final, no more questions, just two comments. One person said that this was the best presentation yet, so congratulations on that accolade.
Thank you very much.
And then thank you so much for your research, and I look forward to getting involved. So hopefully,
awesome.
You'll have a lot of people want to get involved. So thank you so much for joining us. Be sure to contact Jamie at hypoparafoundation.com and visit his booth in our expo hall under Teams on the left-hand side of your screen. Thank you, Jamie, for everything.
Barry, thank you. Thank you so much for everything you guys do for patient advocacy. You guys are the first place people turn when they get Hypopara, and it's scary. It's petrifying. Those ER visits are horrific, the feelings that people have. So what your group does is amazing, and I'm so grateful that you entrusted that about $30,000 in us to move research forward. So thanks a lot, Barry.
Thank you, Jamie. We really appreciate it. Everyone, we have a quick break. We will start back at 10:15 with a session on pediatric hypopara. So thank you, everyone. Thank you, Jamie, and we'll see you soon. Thanks again.